and let's put a polymerase there as well, right? Because at least the polymerase is a leading strand here and doing a lagging strand since it's back this way. So it gets harder to draw, but let's put one there. Topoisomerases, where would they be? Yeah, far on the ends, right? So this is where, this slide set. Yeah. Remember we talked about, right? Topoisomerases need to be downstream of the replication machinery because they're causing, the replication machinery is causing supercoiling as it moves itself through the DNA. Okay. Okay, so what else? Helicase, DNA, polymerase, origin. We did the origin last time. Single strand binding proteins would be after the helicase, yeah, anywhere on this, anywhere where there's single strand DNA. You can imagine if the polymerase is here, then maybe this is already double stranded DNA, right? Because maybe it already have replicated it, just not shown. So anywhere there's single stranded DNA, there's a binding protein that will stabilize that. Primase? Somewhere on the lagging strands, right? So maybe starting, remember last time we picked out the primers that would have to be for at least one of the possible sets of primers for the lagging strand? That primase would be there making those happen. Okay. And sliding clamp will be attached to the polymerase. So if you remember when we started this, like, was that like 3 months ago? It feels like 3 months ago, but I showed you that video of the transcription. You should be able to go back and identify all those things on that video, or there's lots of other videos out there as well, and that might be helpful for you to look at. Okay, so let's forward. All right, so last time we talked about the different stages of replication. We're going to kind of go through that step by step and see if there's any confusion. But you've now, in essence, we have already gone through all those steps in this previous example. So now we're just going to zoom in and look at each of those individually and see if there's questions that exist. But I expect that we should be able to move through this fairly quickly because we've already done all of it, which is just the second round of it. Okay. So primase. Remember that the whole purpose of a primase is to set down an RNA primer that allows a 3' end to be there, right? You need a 3' end in order for DNA replication to occur, and that's what happens here, right? So you can see here, these are RNA polymerases, right? Because they are putting down RNA bases. And so there is a primase that starts the leading strand, but that's not shown here, but primarily what you're seeing here in the lagging strand, there will be several different RNA primers that start the lagging strand synthesis, okay? So this is typically how you see it, right? Successfully opening up the piece of DNA and then laying down new primers and then having this lagging strand synthesis occur. And you need to make sure that you then, this is where the ligase comes in, is to seal this backbone, where the sugar phosphate backbone is nicked at these Ozaki fragments, and that needs to happen. Polymerase 3 extends it. This is the process of replication. Polymerase 1 removes and overwrites those primers, right? Because this is not, we don't want RNA in our double-stranded DNA, right? So we have to somehow remove that. And that happens through another polymerase that specifically will remove and then overwrite that sequence. Okay? Questions on that? So this is the step-by-step, right? Start the RNA primer, extend it using DNA polymerase 3, overwrite it using DNA polymerase 1, and then you have to then have the DNA ligase come in and seal the nicks in the backbone. Okay? And this is all in reference to the lagging strand, right? These steps do not occur in the leading strand for obvious reasons. There is no nicks. There's no, the overwriting still happens. Right? There's a very initial RNA primer for the leading strand that same process occurs, but that only happens once. Questions? All right, 30 seconds. Explain to your neighbor. If there's no questions, we'll move on. Go. All right, questions for me? Okay. Next step. So here's another view of it. There's lots of pictures of this if you want. This is the same content as here. I have it here just as another view for you if that's helpful. At this point, you should be able to identify leading lagging strands, just like we did last time, 5', 3', all that anti-parallel stuff. That should all be in your head one way or the other. Okay. All right. So the other, I want to, before we move into, well, this kind of, we'll zoom out before we zoom back in again. But one of the other things that you need to think about is how does this process actually occur within the cell cycle, right? So what we've been talking about so far is, and this should be familiar, right? We've all seen this before, right? So G1 is where the this process of the cell is actually growing. We are in the S phase when we're talking about replication, right? Is that we're synthesizing DNA. And then we'll go through G2, a small growth phase, and then we'll go through mitosis, where the cell actually will divide these two copies into daughter cells. Okay, so this process of actually going through replication, remember the whole point here is to create a second copy of the genome as accurately as you can. Okay, so, and you also, as you think about our genomes, usually what's not going to happen is you're not going to start at chromosome 1 and then do the whole thing, and then go chromosome 2 and do the whole thing. There's going to be several points where replication will begin, and then you'll kind of go, they'll join up as the replication forks travel. They'll join up to one another. So you can imagine that the cell has to have a very coordinated system of saying, no, we've already replicated that section of the DNA, don't do that anymore, right? Or, because you can imagine once the replication fork goes through, you can't really tell between the new DNA and the old DNA. And so there has to be some way to do that. And that's what this is showing you up here, is that there is a whole host of proteins that's their only job. It's basically a licensing of the DNA saying, we license this part to be replicated, and once that license is used, it doesn't come back on again until you go back all the way through the cycle again. So this is what it's showing, is that this licensing system becomes active in G1, and there's a host of protein complexes that begin to load onto the DNA. They actually help the helicases get to that DNA, and then start to position them so that they can begin to do the work of replication. And then once that process begins, that licensing turns itself off, right? So that makes sure that you only license each section of the DNA once, and the only time that they turn back on again is when you go back through mitosis. Okay, so I put this out here again, is that you likely have heard about replication before, but I want you to see that the levels of complexity just keep on going further and deeper and more confusing. So at least get the fact that that process of loading helicases and beginning to unwind is controlled by a whole host of machinery that is in sync with the sort of life cycle of the organism. Okay. So what this is showing you is the following: is that this process, what happens is that these protein machineries begin to load onto these origins of replication, and you're going to see that what the purpose here is that you have to make a bubble, right? That you have a double-stranded piece of DNA. These proteins are what their job is to begin to make a transcription bubble, or not a replication bubble, right? So this bubble here needs to be opened up so that the polymerase can bind and the helicase can bind, and we can begin. Okay, so what you're seeing is this protein called DNA A will bind to these specific parts. They will actually help to physically melt the hydrogen bonds that exist between the DNA bases and form this initial replication bubble. That will load the helicases, and those helicases will begin to do their work. Polymerases will come on, and the process will start. Okay? this whole process is synced to the cell cycle. Okay? Take a run of this explanation to your friend, and then tell me where I didn't do a good job explaining. Okay? I know this is new material for you, so I expect that there should be questions when you come back. Go.
Yes, it puts proteins that will begin to create the replication bubble, right? The way that you start replication is to put these proteins on, and that's how they do that. So these proteins won't not allowed to bind there until that they get licensed to this process. So the CDK is so that stands for cell-dependent kinases. So these are signals that will turn off this system once you go past this mark in time in the cell cycle.
Turning off the system so that proteins don't bind the DNA?
Yeah, because if they do, then you'll start replication again. Okay. If you start replication in the middle of G2, you're going to have a polyploidy event, and that will be not good.
So what I'm hearing is like licensing sometimes reasons like, for example, if I need to license the replication to that one it recognizes that something's very important to replicate past it.
Close. So this licensing system will help form the protein complexes that will begin the replication process.
Okay.
So you can see that they are loading on the helicases, these DNA, they're beginning to create a bubble, right? So they're setting up replication so it can happen, which requires these proteins. The only way that those what that? Yeah, so bubbles, when the bubbles form with one another, then they will, they're going to hold. You're not just going to have one replication, but you're going to have several replication portions that are going out with one another. Right? But you won't be able to load another polymerase onto that DNA while this is happening.
Okay.
Once everything has been initiated, the system, right?
So like once those are there, like the replication starts to happen, you don't get to start.
You don't get to start again, so you come back around again, right? So the system becomes active. Once you begin that replication process, all of those signals turn off. You can't reset. Replication will not restart at this point. So that at this point,
In that specific bubble, it won't start again.
in the whole cell.
In the whole cell?
Yeah.
So it's only like one piece is replicating at a time, like one bubble at a time?
all the bubbles replicating at the same time. They were all assembled at the same time. They were all made bubbles at the same time. Then they all, once that replication part starts, you're at this point. Once you're at that point, this licensing system is being actively turned off, so that you can't start that process again.
So after G1, there's a couple of little bubbles everywhere that need to be filled.
Yep, and that's exactly what's happening. Replication is going in that process at that point, right? All right, where's talk to me. Other questions, Ben?
What are the active proteins that make the bubble?
Yep. So DNA A is the one protein that's shown up here. So know that I'm picking out the big players here. There is a host of proteins that are involved in this process, and this is highly complicated, highly regulated. But the big players, DNA A is the first one that gets bound. It uses ATP hydrolysis to begin to form the bubble, right? So you can imagine that the hydrogen bonds that exist between the DNA bases, if you're going to break them, you need to input energy. That energy is provided by ATP. Okay, so DNA A begins to create a bubble, shown here, right? These bubbles, and you can imagine also that origin will likely be what base pairs? AT, right? Why? Yeah, it's only double, right? So AT is, we already talked about this, AT has a weaker hydrogen bond, it only has 2, GCs have 3, right? So your origin is likely going to be AT rich, right? And that's exactly true. You can see that pretty clearly when you look into this. So once the bubble is formed, then you start to load it with the pre-replication complex. So the pre-replication complex consists of a helicase, right? So that the helicase can bound there and to begin the process of continuing that bubble as that fork kind of goes out. And then a whole bunch of other proteins. You can see ORC and MCM and a bunch of other ones. But this is the beginning part of, once you have these proteins, that will begin to recruit the DNA polymerase and all the beta clamp, single strand binding proteins, all those other things that you know about. Those will all come and begin to assemble here. And once you begin replication, all of these pre-replication proteins, they go away. And they don't return again until you come back again to G1.
So like this is showing you some image, but it's all happening throughout the entire DNA, just like at one
That's
time.
right. Yep, because the entire genome needs to be replicated. So these are so there will be multiple different origins across the genome that this is going to happen at. If you're looking at a bacterial genome, this is going to happen just at one place, right? At the origin, and that's it. Okay? Questions? Dave?
Does the CDK turn off the protein so like it can't bind anymore? Or does it turn off the sequence? Or like not
Yeah.
allow the sequence to replicate?
Right, so the CDK, you can't do anything with DNA, the origin will always be there. So the CDK is only going to be acting upon the proteins themselves. So it will inhibit and start to break down all the DNA A proteins and the DNA C, which is the part of the helicase. So those proteins will be actively degraded and they're going to disappear and they won't be allowed to be turned back on again. Okay, so all the proteins that were involved in creating this pre-replication complex will be shut down, be deleted, and they won't come back. They'll be actively discouraged. No more transcription of that gene will occur. They're gone. You won't see them anymore. Okay?
So why do the proteins make that bubble and then or the loop and then the bubble forms above it?
Yep. So you're saying about here and here,
Yeah.
right? Yeah. So there's two. You can see that there's a 9-mer there's a replicated. There's 2 sequences that they are the same sequence, and they said they begin to associate. This helps help position the replication bubble at the right place. Is what's going on. So these 9-mers begin to associate, which then allows this bubble to form over here. So a level of complexity that, for the most part, you can ignore. There's cool stuff going on there, but we're going to let that lie for now. Okay. All right. So I zoomed back out. I wanted to tell you about some of the sequencing, but then here's the details, right? So this is now after all of that. Right, so this is all these steps that we've talked about laid out. So can you walk your way through this and with your neighbor? Explain to them the different steps of replication and the proteins involved in those steps, and see which, if any of these places still remain confusing, and then I'll collect questions at that point, and then we'll move on to talk about how you end this process, or some of the other intricacies that are a little bit problematic. Okay, go. What do you got? You got like some sort of YouTube channel? What's that called?
Pre-replication complex on amino acid ventures.
Yeah.
It's symbol and CDK is just an CDK.
Nice. Nice. small little dots. We can go with purple, the purple hourglass. You see the hourglass? Bottom.
Oh my gosh, I'm not there yet.
Oh, check. You guys good over here? Yeah, they have, there you go.
It's just another name, just topoisomerase.
Yep.
And if you reference it, or you would just say topoisomerase. Okay.
This technique is so.
If that's still confusing, go back to the video that I started with. If you search DNA replication, that video is one of the most popular one that people put up there. Okay, so this is the same thing, just drawn differently. Okay. So what we haven't talked about a lot is this beta clamp. So I just want to show you how that thing works and show you where that kind of gets in. So looking at that, the replication complex, like with the polymerase being stationary and the DNA gets looped out and around, the tau protein is what holds together the two different polymerases. Okay, and that tau protein has a couple different unique sort of functions. It's an area of active research, especially around Alzheimer's. So there's some interesting things about what tau does. The other thing that you see is that this is your beta clamp loader. So here's your lock nut washer that I was talking about. This is the beta clamp. Notice that the beta clamp loader gets added on right to the front side, I guess you consider, like, so this is a leading strand, or depending on your perspective, strand is moving this way, so the beta clamp is on the side that just completed the replication, or I guess the back side of the polymerase, or the front side of the new strand, however you want to call that. But this is holding the DNA to the double strand itself. Okay, so that's the job of that. And there's a protein that will open up that beta clamp, and then it will attach it and put it back together again. So opens up and then seals it around so that this polymerase can't fall off the DNA. Okay, so this is the only job of the beta clamp is to do this work of holding the polymerase on. Okay. Questions on that? Yep, Allie.
You said with the beta clamp is that one thing. What is the Y clamp thing?
Yeah, this is the protein that will physically load it onto the DNA itself. Okay. So as this clamp is going to be completely circular, it has to be a protein that will physically open it up and then load it and then hold it and put it back together again. Okay. And this thing has to be active because, at least in the lagging strand, it has to do this every time that there's a new fragment, it has to put another clamp on. So this thing's going to be quite active on the lagging strand. Only does it once, obviously, for the top strand. Okay? All right. So process should be straightforward. Now what we need to talk about is error and also ends. Okay, so the whole point of this process is to make sure that you make no mistakes. It's a really high bar, right? If you think about the amount of DNA that exists in a cell, we're talking about hundreds of thousands of bases. And so these polymerases cannot make even one mistake. And they typically don't. They make one mistake usually in about 1 million base pairs. So the success rate of these polymerases is outstanding. So the question is how? There's no way that something could do it that well and not make mistakes. So the first thing is the following. So if you look at just a strand, so here's your free 3' end. What happens is the DNA polymerase is bound to that double strand or that growing strand of DNA, and all these nucleotides are existing in solution. Okay, so what happens is the polymerase is bound there, and these DNA nucleotides are diffusing in and out of the active site, just select and seeing which one matches up. Okay, so you can imagine we know that the A and T will bind together really well, and that will be a nice, happy marriage. And so the vast majority of the time, what will happen is that A will diffuse in there, will bind, things will be good, the DNA polymerase will move on to the next one. Okay, but it's not like the DNA polymerase is taking something of it and then like putting it in there, right? This is all based on diffusion. Like all those nucleotides are existing and they're coming in and out of the active site, and once that base pair is a good match, it then moves on to the next one. You can see that at some point, you could have a scenario where just because the G is in this soup, it will diffuse into the active site and will try to pair with the T. What will happen in that scenario is that the DNA actually will be physically deformed. The T and the G do not hydrogen base pair or hydrogen bond to one another, and so it will be bulged at that place, right? Is that it will know that this is incorrect by the physical structure of the DNA. When it sees that, notice that it has already formed. Notice that the sugar phosphate backbone has already been formed, right? So the DNA polymerase already put this G in, but it will notice that it's not hydrogen base pair as you would expect. And so what happens is that it takes and uses this. It has a 3' to 5' exonuclease activity. So it means it can go in reverse and chew bases back this way. So it moves in a 5' to 3' direction. So it replicates in a 5' to 3' direction, but it also has a reverse mode, a 3' to 5' exonuclease activity where it can chew bases back off again. So this is one of the first ways that the error rate is so good, is that the DNA polymerase checks itself. Every time it does its job, it says, how did I do? All right, looks good, I'll move on. And if it makes mistakes, it says, oh, let's go backwards, take it out, let me try again. And then it goes forward again, hopefully getting the right one. Okay? So this is the first level of error correction that the polymerase has. Okay? Explain this to your friend, and I'm going to give you the next level after this. Okay, go. Yeah. So I'm going to give you the short answer, and then I'm going to tell you to go back to the and make, because I think it will click when you see it. But if you look at A and T, there will be 2 hydrogen bonds that occur between them, and when those hydrogen bonds occur, they will bring it in line. If A tries to bind with G, if you look back, it's purine and pyrimidine, right? So those bases are wildly different. One's a double ring and one's a single ring. If you have 2 double rings here, what will happen is that it will create a bulge. There's physically not enough space for these 2 things to come together, right? And when that happens, that bulge is what's being sensed by the polymerase saying, whoops, that's not right. So when you have this mispairing that's occurring, it's not going to create this nice linear double helix pattern that the polymerase is expecting. And when it physically senses that bulge, that's when it exonucleates it. It also, you should know, so this is the other trick, right, which I didn't really talk about, but the parent strand, it knows where the parent strand is, and it knows where the newly strand replicate is as well, right? So it has to take out the right one, right? So it makes sure it takes out this one and not this one, because that would cause all sorts of problems, okay? So the template strand exists in the active site. The exonuclease activity is on the top end of that of that polymerase, and it's going to take out the newly synthesized strand that's wrong. Ben?
If it's already past the base, how does it recognize that it's...
If it's passed, it's over.
No, I mean, like, as soon as it
Oh.
leaves it, how does it recognize? Because isn't it technically sort of behind it? Or is it almost like a momentarily, as soon as it lays it down, it's like...
Yeah, so this is still in the active site. It just put it in, and it's sensing it right now. But if it moves on to the next base, there's another process that will catch that. That mistake is now in stone, and something else is going to have to fix it. But it's not going to do the polymerase.
Okay, I get that there's like a bunch there, but like how does it just like physically sense it?
Yeah, so are you going to take biochemistry with me next fall? That would be wonderful. So can I just, well, more of them, but let me tell you, so the short answer is that if you look at proteins, especially if the DNA polymerase protein is a massive protein, it's like hundreds of kilodaltons large, the active site is a very small physical space. So like in biochem today, we're talking about hemoglobin and how hemoglobin binds oxygen. The difference between hemoglobin binding oxygen and carbon dioxide, so you know that you can kill yourself if you put a high carbon monoxide into your, right? The reason why that is that carbon monoxide, C and O, and O and O, they're different by, like, we're talking microns. Like, they are so small different. But the fact that carbon monoxide can stand straight up in the active site and oxygen can't, that difference is what that protein is recognizing. So these proteins are incredibly fine-tuned to what they do. They very easily can see this bulge because it's only slightly different than what it should look like, but it's because that space is so small and these things are so precisely tuned, it's like night and day. Like, oh, that's clearly a mistake. We have to undo that. So you'd be surprised how tightly tuned these proteins are. They're amazing machines. Yeah.
No,
that question?
Sure.
Okay. All right. So we talked about. So remember, the purpose is need to be highly accurate in this process. This is one way that happens. Okay. Here's the other way, right? So what happens if the DNA polymerase is having an off day and it completely blows past a mistake? That happens often. It happens sometimes as well. And so then now there's another layer of defense, and that is a whole host of proteins that all that their job is to scan the DNA at this point, at replication, and see if there's any mistakes. Because once you go past replication and you start to have those cells divide and have these new DNA into these new strands, it's too late at that point because now that strand looks like it was any other strand. So there's a very small window at which point, right after replication, you're checking the DNA to see if it's okay before we move on to division. Okay? So I'm just going to give you genetic, in more classes we'll talk about this in greater depth, but I want you to have the first run about what's happening. Okay, so what you see here is happening is that here's your parent molecule. It has a GC base pair. You replicate it. The top strand does a good job, GC base paired. But on the bottom strand, notice that there's a mistake with an AG mispair, and the polymerase missed it. Okay, so the question now is that now you have a piece of double-stranded DNA that has an A and a G, and you have to figure out a way to say which is the right base and which one's the wrong base, right? Because now they look like DNA strands, and so you have to figure out which one's parent, which one is daughter. And so the way that this happens is that you can imagine that you don't have strands of like red and yellow in the cell, right? You can say, oh well, it's clear because it was a yellow strand, so we're just looking for yellow. But sorry, that's not the real life application here. So the way that the cell figures this out is that if you're not aware, methylation is adding of a methyl group onto DNA. This happens to DNA as a natural consequence of being in the cell. So you would expect that all of the old DNA should be methylated at the marks along DNA that they are. And the newly stranded DNA hasn't been in the cell long enough to be methylated. And so this is one of the ways that the cell can recognize old versus new. So what happens then is that you have to then go back and say, all right, which of these is the methylated strand? Okay, this is the methylated strand. That means that this is the error. So let's take that out, and then we can then fix it. Okay? Back to where it should be. If you don't fix it, now you have a point mutation. Right? Is that now when this new piece of DNA goes under replication the next time, now you have this AT base pair, which should be a GC. Okay? And at this point, once you're here, there is no going back, right? You can't remember from 2 generations ago that you used to be a GC. This is now a mutation that may or may not have a consequence for the cell. Okay, Zach?
So what is looking for the methylation?
Yeah. So do I go on to that or not? Yeah. So let's talk. So let's go forward, then we'll pause. Okay. So here's what the repair looks like. This is called mismatch repair. This is a complex of proteins. Its job is to scan the DNA and look for mismatches. Okay. So mismatches that the DNA polymerase has forgotten or has overlooked. Okay. So you can see again old template, new template. With colors, obviously the cell doesn't have those colors, but what it does have is those methyl marks. Okay, so at certain sequences on the DNA, there will be methylation tags. So at CTAG, the guanine gets methylated. You put a methyl, a CH3, on the DNA. Okay, so what needs to happen then is that if there is a mismatch, so notice that one of the yellow protein, which isn't yellow, but the yellow protein binds to mismatches. That's its job. Or I'm sorry, the brown protein finds mismatches. So the brown protein's job is to literally be on the DNA and scan it and just go up and down the DNA, back and forth, until it finds mismatches.
What is the brown DNA, or not DNA, its name?
So mute. We aren't, but you want me to tell you?
Yeah.
So this is mute M. So that protein's job is to scan the DNA and find mismatches, and it finds mismatches the very same way that the polymerase does. It looks for bulges where the base pairing is not good. Okay. So this mute M protein, that's what its job is to find the mismatch. But at this point, it has found a mismatch, but it doesn't know which strand to cut. So then what has to happen is that its partner, mut T, in yellow, has to find a methylation tag. Wherever that so the nearest methylation tag that exists. It will scan until it finds a methylation tag and it will bind there. And then this blue thing, I think this is mute. I don't remember what this one is. This is the matchmaker. The matchmaker brings these 2 complexes together. And when those complexes come together, that activates the exonuclease activity of these proteins. And this yellow guy will cleave the unmethylated strand and will eat all of the DNA until it comes back to its partner. Okay? So basically what's done is that you get rid of all of the new daughter strand from the methylation mark to the mistake. Okay? So notice that this is a very expensive process. You have made one mistake, and you're chewing up 3, 4, how many of your 100 base pairs it gets back to in order to get to methylation mark. And then you can imagine that DNA polymerase 3, the one that does the short repair work. DNA polymerase 1 comes in here and then does patch repair and puts down all new bases that you just chewed up. Hopefully this time doing a better job than the previous one. Okay? So notice that this is now your second line of defense. The polymerase itself was checking itself all the way throughout, and now these proteins are double checking the polymerase's work. So if there's any mistake that the polymerase makes, hopefully these complexes catch that. Right? And this is only one of a host of DNA repair mechanisms that exist that are out there.
Which one's the link to like the 808? All of the ones that you errors?
The yellow one is going to cleave and then remove. Jade.
What's the blue one?
The blue one is the matchmaker. It brings those 2 proteins in contact with one another, and only then will the exonuclease activity be activated. If you have a mismatch and you have found a methyl mark, at that point when you have both of those ingredients, now you're ready to excise that DNA.
You said that this is only like one of many ways
Yeah.
to fix it. Or will we get into like
No, genetics.
so we only
Yeah. So I'm peeling back layers slowly, right? I want you to understand that there is a host of things that the cell has to do in order to keep the DNA as secure and as the fidelity remains high, right? That we don't make any mistakes. This is critical.
Since it is so like expensive that it gets rid of so many, is it very common? Is there other methods that are better?
No, this is our mismatch repair system. There's other mechanisms to deal with other mistakes or other problems, but this is how we deal with mismatch. So if you have UV damage, like if you have too much sun, that causes thymine dimers, if you've heard of those. Basically, two thymines being covalently bonded next to each other this way as opposed to this way. A whole nother problem that another set of cells have to deal with, another set of proteins have to deal with. Okay? All right, have a great weekend. We will finish this.